Here we are again. Today's lesson is on cell anatomy, the structure of the cell. So just a little briefing. Human body contains approximately 75 trillion cells, of which there's over 200 different types of cells. Some things that are common about cells: they obtain nutrients, they get rid of waste, they provide shape and integrity, which is maintained by internal contents and the surrounding membrane. Their functions and characteristics are determined by the genetic material, and most cells are capable of undergoing cell division. Now, what is a cell, right? We consider the cell the basic unit of life, and essentially, a cell is surrounding a bunch of chemicals and molecules, and they are separated from the outside world by a plasma membrane. Plasma membrane is made primarily of lipids, a special type of lipid called a phospholipid that is can interact with both water and with other lipids. And so it creates this barrier that separates the inside of the cell from the outside of the cell. And that's really what the beginning of life is creating this internal environment that's maintained by the cell. And so the cell has evolved over many millions of years to what the human cells look like today. Cytology, cyto is the prefix for cell. This is the study of cells and uses mostly microscopy, or the use of microscopes. So cell sizes are measured in micrometers. This little Greek letter mu stands for micro, and micro means 1/1,000,000th. So a micrometer is 1/1,000,000th of a meter. That means there's 1,000,000 micrometers in 1 meter, right? A meter is like, so big. A centimeter has 10,000 micrometers in there. So within the human body, we have lots of different cells, ranging from the smallest, like red blood cells, to some of the bigger cells, the oocytes, or the female egg, which is about 120 micrometers. Usually we measure these across or by diameter. Here are some basic ways that we look at cells through different types of microscopes. We, in our class, use primarily the light microscope. The light microscope shines light up through a thin section of material, and we can see some contrast. Oftentimes, we stain our specimens with certain dyes that are going to allow us to see the structures more clearly. Some other types of microscopes are the transmission electron microscope, or the TEM, or the scanning electron microscope. And actually, Delta College has a fantastic electron microscopy pathway. And so that if you like taking really cool pictures of really small things, that might be something to check out. But getting to the meat of our time here is we want to look at the anatomy of a cell. Remember that the word anatomy means structure. So this course is all about structure. And so we can think about functions a little bit, but really what we're concerned with are structures. And when we look at a cell, especially under a light microscope, there's going to be a few things that we want to identify. So first is we should be able to see the outer rim of a cell. Let me get my pointer on here. The outer rim of a cell. And again, that is surrounded by the plasma membrane. And that that allows for the external environment and the internal environment to be separated from each other, and that allows life to occur. Um, the biggest, most prominent thing that we will see in a cell is the nucleus. And so the nucleus is depicted in this um, artist rendition, like this right here. And the nucleus contains all of the genetic material of the human cell. So all of our chromosomes, which are made of DNA and protein, are all contained within the nucleus. And so that's usually prominent when we find a cell, when we're doing histology, the study of tissues. We're going to look for those nuclei and we're going to look for their shape and see where they are. Some other prominent features in here. These little guys are called mitochondria. Mitochondria, sometimes called the powerhouse of the cell. Mitochondria are the organelles, the little organs of the cell that do a reaction called cellular respiration. And this is where glucose that we eat and oxygen that we breathe are combined together in a really explosive chemical reaction that releases a lot of energy, and that energy is trapped as molecules of something called ATP. And that's how we use energy in our cells. That process all happens here. The waste from that process is water and carbon dioxide. So, we breathe in oxygen, and that goes to power the reaction, cellular respiration that takes place in the mitochondria. And then we breathe out carbon dioxide. That carbon dioxide is coming from that reaction here. Let's see, what else can we point out in here? All cells are going to have these little structures, these little dotted structures around here. Those are called ribosomes. And those ribosomes take the instructions from DNA and they turn them into proteins. There are some other features in here. This kind of blobby looking stuff is called the Golgi apparatus. This extra blobby looking stuff is called the endoplasmic reticulum. All of the organelles are bathed in a liquid, kind of a soupy liquid called the cytoplasm. And then some cells, not all cells, will have protrusions called microvilli or cilia or flagellum or microtubules. Those are extra features that some cells will have. And then other things that are pretty important here is these little membrane-bound bubbles that kind of float around through the cell and transport things like proteins and waste either throughout the cell or allow it to go to the outside world. Those are called vesicles, or as my Canadian professor used to call them, vesicles. Other than that, I think there are a few other structures that you should be aware of and that you should study and know, but I just wanted to highlight those major ones. And that is all we're going to talk about with cell anatomy. Hope that was helpful. See you next time.